Hi, I'm Barbara. I'm a natural sciences student at the University of Cambridge, and this summer I carried out a project that looked into the duration of transcriptional memory in Arabidopsis thaliana. So, in the context of plant pathology, the idea of transcriptional memory means that once plants have been primed, so exposed to a pathogen the first time around, then the second time around that they say it, their response is much faster and stronger. And this is underpinned by changes in levels of expression of particular genes, which are called memory genes. And I think the first reason I applied for this project was really that this idea seemed very, very cool to me and I wanted to know more about it. And the second reason was that I wanted to gain more practical skills and also get an insight of how working in a lab really looks like. In terms of what my project involved, I had both a wet lab part and a computational part. So in the wet lab part, I was performing QRT-PCRs to compare the levels of expression of particular genes that I chose to investigate. And in the computational part, I was using the RNA-seq dataset that was collected by my supervisor to identify genes that were differentially expressed between primed and naive plants. And also, I compared our dataset to other datasets that were investigating the kind of transcriptional response to exposure to a pathogen. Additionally, I performed a ROS assay, which was looking at priming at this sort of level of immune response, which was also interesting and added to my project and how I could relate what I was seeing at the level of transcription to the immune changes. And I think what I really enjoyed is that I just had many different parts and I was doing multiple different experiments. And this meant that I was learning loads of new skills and not just repeating the same thing every time. Um, and in terms of what I learned, I definitely learned loads of useful techniques in biochemistry like PCR, QRT-PCR, or RNA extraction. The computational part definitely massively increased my confidence in coding, especially in R and command line, and also really inspired me to delve into bioinformatics more deeply and also perhaps develop in this area in the future. Given how much I enjoyed my internship, I definitely would want to further my research skills um, and learn more about plant pathology. And also, it helped me to choose my subjects for next year, and I'm really looking forward to be doing plant sciences then. Overall, I really enjoyed this internship. It gave me so much opportunities to learn more about research. I'd like to thank my supervisor Jake for being very, very helpful and supportive and encouraging throughout this project. All lab members, in particular, for incredible help on different experiments that I was doing. And of course, BSPP for providing me with this opportunity.